Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Aisyah binti Akbar Ali, metric number 23DBT19F2018. The title of our research is Comparison of Nutrient Content in Animal Feed Produced via Silage and Anaerobic Digestion. Introduction. Malaysian livestock industry is an industry that contributes approximately 16.1% to the total gross value added of agriculture sector. It plays a role to provide lucrative employment and also produce and supply animal-based proteins such as meat and milk. It can further be classified into two categories, which is non-ruminant and ruminant sector. Ruminant sector consists of beef and dairy cattle, dairy buffalo, sheep and goats. Due to not being able to fulfill local demands, Malaysia still imports beef, mutton and dairy products from other countries such as India, Australia and New Zealand. Some of the factors that contributes to this is lack of land resources, high feed price, cheaper import substitute and etc. One of the factors that we would like to focus is the quality of animal feed. So a low quality feed will cause poor gut health, reduce digestion rate, affect growth rate, and in some cases also cause disease and inflammation, which will affect the sector badly. So in this project, we will be producing two types of animal feed via two methods, which is silage and anaerobic digestion. So the main difference between these two methods is that silage consists of three phases, which is the aerobic phase, anoxic phase and anaerobic phase, and we'll be using microbes that can survive all these conditions, whereas anaerobic digestion will contain only anaerobic phase and will use microbes that only survive that condition. So the two type of animal feed will be used is napier grass and oil palm waste. So the reason we chose oil palm waste is to reduce the production cost and also to reduce waste. So the purpose of this research is to in hope to create a low cost, high quality animal feed with high nutrient content. We also hope that we can make a good quality feed more accessible to poor farmers and to produce an animal feed which is more environmental friendly and reduces agriculture waste. Objective. So there are three objectives corresponding to the number of teammates. So our first objective is to investigate the preferences of farmer on unprocessed feed versus processed feed via a survey. Our second objective is to analyze the carbohydrate, protein, lipid and microbial content in animal feed produced via silage. And our third objective is to analyze the carbohydrate, protein, lipid and microbial content in animal feed produced via anaerobic digestion.
So now I will explain about literature review. Malaysia's GDP declined 5.6%, influenced by declining in all sectors economy compared to positive growth 4.4% in 2019. The contribution of agriculture sectors is 7.4% in Malaysia's GDP. The sector's percentage increase fell to 2.2% to 2.0% the previous year. The causes is commodity subsectors, to be exact oil palm negative growth, which is 3.6% while 2019 1.5%. Oil palm actually the largest contributors to agriculture value, accounting for 36.9 billion, like 37.1%. Livestock in Malaysia are ruminant and non-ruminant. Ruminant consists dairy cattle, beef, dairy, buffaloes, sheep, and goat. The ruminant are still raised in a small scale and not meet a local demand. Malaysians need to import the stock from abroad. One of the factors is high feed price. What fermented food have more available simple nutrient and make nutrient absorption become faster and efficient. The food can be safe, self-made, and it can cut costs feed. It can produce higher yield and we don't have to depend on foreign country anymore. Gut health relates to nutrient absorption and infection are kept out of digestion tract. Gut health defined as strong barrier between digestion tract and bloodstream. Various disorder will occur if the digestion tract exposed to pathogen like laminitis, liver abscesses, acute interstitial pneumonia, and inflammation. A good gut health help to prevent these diseases to occur, and beneficial bacteria in fermented feed can change the microbial environment in cow intestine and also make the feed easy to digest and absorb. Beneficial bacteria can also increase disease resistance, reduce the occurrence disease, and antibiotic usage. The major process of silage is aerobic fermentation, stable, and fade out. The difference between silage and anaerobic digestion is anaerobic digestion relies on anaerobic microorganisms to digest. Anaerobic digestion avoids any contact with oxygen. So, this study was carried out to determine the differences in nutritional content in two different processed food, which is anaerobic digestion and silage food. The hypothesis for this project is farmers prefer processed feed due to the high availability of nutrient content in comparison to unprocessed feed. The carbohydrate, protein, lipid, and microbial content in the feed produced via silage is less available. The carbohydrate, protein, lipid, and microbial content in feed produced via anaerobic digestion is higher and more available. There are three methods that we will use in this project, which is survey. We will ask farmers on their preference on animal feed, whether it's processed food or unprocessed food. Second, feed preparation to prepare silage and anaerobic digestion food. And lastly, nutrient analysis. We will analyze the content of nutrient in the food. Okay, for survey, we will conduct by interviewing sample according to convenient sampling method. The sample is taken from farmers that easy to reach and easy to contact. The farmers will be interviewed on their preference on animal feed, whether it's processed food or unprocessed food, and the reasons behind it, like the understanding of farmers in the two types of food, the cost, and effect to the animal. A pilot test will be conducted to analyze the data.
Feed preparation. So two types of animal feed, which is the Napier grass and oil palm waste, will first be gathered and collected. Then both feeds will be produced via two methods, which is via silage and via anaerobic digestion. So these are the following materials needed to produce 1 kilogram of silage. First, 1 kilogram of Napier grass and 1 kilogram of oil palm waste will be chopped and ground into fine pieces. This is done in order to increase the surface to volume ratio and to reduce time. Then the grinded sample will be added into a container and hot water 80 degrees Celsius will be added in a ratio of 1 to 3 for hydrothermal pretreatment. Then all the materials listed above will be added into container and mixed until a slurry is formed. Then the pH and temperature of the mixture will be taken and 100 gram of sample will be taken out to analyze the initial nutrient content. Then the container will be covered with polythene sheet to prevent any water content and to prevent airflow. The container will be left at room temperature for 3 months, and after 3 months the nutrient content in the sample will be analyzed. As for anaerobic digestion, the process is same as silage making, but a different bacteria which only survives in anaerobic phase will be used, which is the methanogen. And the experiment will be conducted in vacuum condition to prevent any presence of oxygen.
Bismillahirrahmanirrahim. My name is Muhammad Ali Hassan bin Nur Rahman, 23 DPT 19F 2008. I will talk about nutrient analysis. In our proposal, comparison of nutrient content that have in this animal feed has 4. The first one is carbohydrate, the second one is protein, number 3 is lipids, and the last one is microbiome. Okay, the first one, to find the how much carbohydrate in animal feed, our product is we use phenol sulfuric acid menthol. For the protein content, we use laundry menthol to find how much protein in this product. The third one, for the lipid, we use light and dire menthol. Last one is microbial content. We use quadratic stirring. For biochemical tests, have 4 in microbial content. The first one is catalase test. The second one is indole test. Number 3 is VP test, and the last number 4 MR test, and the last one is gram staining. For carbohydrate, the first one, step 1, place 200 milligram of glucose in a test tube and make up 100 ml using distilled water. This will be used as the starting mark of the serial dilation to prepare the standard curve. After that, prepare 3 test tubes containing 50 ml distilled water and serially dilute by using 50 ml of the standing mark as start. Next, pipette 1 ml of the standard solution into the test tube and 0.5 ml of 5% phenol solution along with 2.5 ml of concentrate sulfuric acid. And then vortex and incubate in water bath at 30 Celsius for 30 minutes. Measure the absorbance at 419 nm and plot the standard graph. Repeat procedure 3 until 5 using supernatant of phase sample. The last one, record the carbohydrate concentration using the standard graph. Procedure for protein. The first one, place 0.5 mg of PSA working standard in a test tube and make up 100 ml using distilled water. It will be used as starting mark of the serial dilution to prepare the standard curve. After that, prepare 3 test tubes containing 50 ml distilled water and serialize dilute by using 50 ml of the starting as a start. Prepare the test tube with 100 ml distilled water that will serve as blank, and add 4.5 milliliter of region 1 and incubate for 10 minutes. After that, incubation at 0.5 milliliter of region 2 and incubate for 30 minutes. Measure the absorbance at 616 nm and plot the standard graph. Estimate the amount of protein from the supernatant of the process waste from the standard graph. For the procedure of lipid, step 1 is take 4 grams of waste sample and mix with 10 ml of chloroform and 20 ml of methanol. Stir and mix well. And then add another 10 ml chloroform and mix well. After that, add another 10 ml of distilled water and mix well. After that, filter the solution using Wattman number 1 filter paper. After that, transfer the liquid part to graduated cylinder and wait for it to separate into two parts. The first one is chloroform pass and methanol pass. Record the volume of chloroform pass and remove methanol pass using a pipette. And then wick an aluminum dish and transfer 5 ml of chloroform into the aluminum dish. After that, place the aluminum dish in drying oven at 105 Celsius for 50 minutes. The last one, cool the aluminum dish in the desiccator and wick. Then conclusion for lip content is followed by mass of lipids equal WCX V chloroform divided by V aliquot. For the culture mentor quadrant strict for microbiome, we have 5 biochemical tests. The first one is catalase test. For the result, bubble positive result for the presence of catalase. The second one is indole test. Indole test will observe for the presence or absence of ring. Okay, number 3, VP test. VP test. A cherry red color indicate a positive result, while a yellow-brown color indicate a negative result. The test depends on the digestion of glucose to acetylmentylcarbinol. Okay, number 4, MR test. MR test for the result observe for the color change in the broad medium. For gram staining for result, a dry, blow dry, and observe under microscope to see the gram positive or gram negative.
Expected results. So the first one is farmers prefer processed feed due to high availability of nutrient content in comparison to unprocessed feed. The second one is the carbohydrate, protein, lipid and microbial content in feed produced via silage is less available. And the third point is the carbohydrate, protein, lipid and microbial content in feed produced via anaerobic digestion is higher and is also more available.
Budget for this proposal to make the animal food in 100 plus. The example of item, neighbor grass for silage nutrient, main nutrient, food for animal, or plant waste, molasses, salt, urea, and 46%, the calcium phosphate, mineral mixture, bacteria, and metagen.
For planning for this project, the topic selection was begin at September until October 2021. Then, research proposal was framed and prepared from November 2021 to January 2022. We will make the survey on farmers on February to March 2022, and feed preparation expect to be done in 3 months, which is March to May 2022, and the nutrient analysis will be conducted on June 2022. The project expected to be complete by the end of the June 2022.